Hello guys, in this video we're going to go ahead and move on to topic 3 of the 4.1 notes. In topic 3 we're going to be looking at different distances of cell communication. And so far in topics 1 and 2 we've talked about these signal transduction pathways and what happens to cells when they receive a signaling molecule and the events that occur inside of the cell. In this topic we're going to kind of look at, we're going to zoom out a little bit and look at just overall in general these different ways in which cells are communicating with other cells that are either close to them or far away, and how that takes place. And we're also going to be looking at lots and lots of examples, or a handful of examples of these different distances of cell communication and what it kind of looks like in different situations. And so like I said, cells can communicate with each other either with other cells that are either very close, and we call that short distance communication, or cells that are very far away, which would be a long distance. So we're going to start with close cells that are close to each other. There's one type of signaling that involves cells actually coming into direct contact with one another, and that's called juxtacrine signaling. And that is whenever you have a situation where one cell is communicating with another cell, and it relies on them being in physical contact with one another. So one example, so this is going to involve no releasing of a signaling molecule. So in this situation, the cell that's sending the signal is not actually going to secrete the ligand, the signaling molecule, that will then bind to the other cell. In this situation, there is no secretion of a signaling molecule because it's going to rely on them being touching each other, basically. And so one example that we've actually seen before this semester is how in plant cells and animal cells there's this certain type of cell junction. There's these different types of cell junctions that have these open channels between adjacent cells. And so in animal cells they have these things called gap junctions, where two different cells are next to each other and they're joined together, but there's these open channels that allow molecules to diffuse from one cell to the other cell without having to be released from the cell. And so in a lot of situations that can involve signaling molecules from this cell going directly to the other cell without having to be released. Same thing with plant cells. Plant cells have something called plasmodesmata, which is a type of cell junction between adjacent plant cells where they have these open channels between the cytoplasm of both of those two cells. And so that again can allow signaling molecules like ligands to directly diffuse between those cells, which can trigger certain forms of communication between those cells. So again, that's through direct contact, like those cells are physically in contact with one another. Another example of juxtacrine signaling is when a cell has these certain molecules embedded on its cell membrane that can bind directly to receptor proteins on another cell. And so in this picture right here, you can see there's a cell that has these certain membrane receptor proteins, and the signaling molecule that's going to bind to that receptor protein is actually embedded on the cell membrane of another cell. So this could be like some kind of a protein or some other molecule that's displayed here on the cell membrane that's able to, through direct contact, when these cells are in direct contact with one another, that surface molecule can bind to this receptor protein on the cell right next to it, and that can then trigger this receptor to become activated and things to happen. So that's called juxtacrine signaling. Juxtacrine signaling is whenever two cells are in direct contact with one another, communicating with each other. And in these situations, there's no secretion or release of a signaling molecule. And then other types of short distance communication. So it is possible for cells to also communicate with cells that are very close by without direct contact. And so there's a couple examples called autocrine and paracrine signaling, where cells are going to release a signaling molecule. They're actually going to secrete a signaling molecule, a ligand, that's then going to bind to receptors on a nearby cell. In autocrine signaling, we have a special situation where a cell is actually communicating with itself, basically. So it's releasing signaling molecules that can then bind to receptors on its own cell. That's called autocrine signaling. An example of that is like in lots of different types of cancer cells, they can release these signaling molecules called growth factors that can then bind to their own receptors, and then actually those receptors trigger signal transduction pathways inside those cancer cells that then tell those cells or cause those cells to start dividing faster and dividing more. So that's an example of autocrine signaling. Anytime a signaling molecule is being released and then binding to receptors on the same cell that released those signaling molecules. And then there's paracrine signaling. Paracrine signaling is when there's a cell, again, releasing a signaling molecule, and that signaling molecule is going to, through simple diffusion, is going to diffuse nearby and bind to receptors on nearby cells. So like in this picture right here. And there's lots of examples of that where cells are releasing signaling molecules that are going to bind to receptors on lots of cells that are in close proximity to this cell, where that signaling molecule can just diffuse away and bind to those receptors. That's called paracrine signaling. And so now what I want to do is I want to go over a few different very common examples of this type of short distance communication that come up a lot. Some of these are a little tricky, these examples, so I'll try to explain them as best I can. One of them is morphogens that are involved in early embryonic development. So very early on when an embryo is developing, basically when it's starting off as just a clump of cells before it starts becoming or morphing into like something that looks like a real living offspring. But at the very early stages of embryonic development, there's these signaling molecules that are called morphogens that are really important. And so what happens in this situation is in this clump of cells, so imagine very early on, you have a fertilized egg, and that fertilized egg has now divided into a clump of cells. And then there's going to be some of those cells that are these morphogen producing cells. So there's certain cells that start releasing the signaling molecule called a morphogen. So morphogens are these signaling molecules being released by some of these very early embryonic cells. And those morphogens will then diffuse away to all these other cells that are in this clump of cells, right? So there's some that are really close to this cell, there are some that are further away. But what's going to happen is there's going to be a concentration gradient that's produced. So if we're producing morphogens, like if this cell is producing morphogens, who's ever closest to that cell is going to be exposed to a very high concentration of morphogens. And the cells that are further away from that cell are going to be exposed to a lower concentration of morphogens. And then what happens is these other cells, these other embryonic cells, they have receptors for morphogens. So morphogens, these signaling molecules, can bind to these receptors on all these cells. All these embryonic cells have receptors to these morphogen molecules. And depending on how close you are to the person who's releasing those morphogens, to this cell here who's releasing those morphogens, you're going to have more or less of your receptors becoming activated. And so like if you're really, really close to this cell, you're going to be exposed to a high concentration of morphogens, which means very likely a lot of your receptors are binding to a lot of your morphogen receptors are binding to that signaling molecule, the morphogens. Whereas if you're further away, it's less likely that you have maybe a lot less receptors binding to those morphogens. Now, and then so what's going to happen is depending on how much of what proportion of your morphogen receptors are becoming activated, it's going to trigger different cellular responses. And in this case, it's going to cause certain genes to turn on in these cells. And so just to keep it kind of overly simplified, let's just say if you have like a huge amount, close to like all of your morphogen receptors becoming activated, that's going to trigger a certain set of genes that then tell this cell to start developing into a certain type of cell. So maybe this will trigger and turn on genes that tell this cell to start developing into muscle cells. And so now this guy is going to turn on, the cellular response that's going to happen is there are certain genes in the cell that are going to turn on. And let's say it's the genes that are needed for this cell to become a muscle cell. And then he's going to start becoming a muscle cell and dividing and making more muscle cells that are going to form muscle cells in the developing offspring. And then this guy right here, his morphogen receptors are like half of them are being activated. And let's say that triggers a different set of genes now turning on. So because half of your receptors are becoming activated, that initiates a slightly different signal transduction pathway that leads to a different set of genes in this cell turning on. And let's say it's the genes that are needed for this cell to develop into a nerve cell. So this guy's going to now start turning on genes that allow this cell to differentiate and become a specialized nerve cell. And it's going to start forming your nervous system and your brain and the nerves that travel through your body, and all the cells that make up those tissues, the nerve tissue inside of you. And then this guy, he's far away, so he doesn't have a lot of morphogens binding to him, and so that might trigger a different signal transduction pathway that causes another different set of genes to turn on. Let's say it's the genes that tell this guy to become a skin cell, and so now he's going to start turning on genes that allow him to start developing into a skin cell. And so these cells, basically what's happening here in summary, they are each being, there's responses in these, certain genes are turning on in these cells, and the set of genes that are turning on is dependent on the concentration of morphogens that they're exposed to and the amount of receptors that are being activated in these cells. So you can kind of see here, here's a graph showing, like, if you're really close to the source, the person who's making morphogens, you're going to be exposed to a high concentration of morphogens, but as you're further away, you're going to be exposed to a lower and lower concentration. And depending on the concentration of morphogens that you're exposed to, that's going to trigger different outcomes in those cells. In this case, it's different sets of genes turning on, so that this clump of cells can now start, the cells can start differentiating into different types of cells, so we can actually move along through development and start forming an offspring here. So that's the morphogen example, and this is another good picture that kind of summarizes how these cells are going to respond differently depending on the concentration of the signaling molecule that they're exposed to. So that would be an example of short distance communication. So we would call that paracrine signaling. So a guy is releasing signaling molecules who's binding to receptors on nearby cells. Okay, so that's paracrine signaling. Another example that's also kind of confusing is something called quorum sensing. This is something that certain types of bacteria are able to do. And it's basically a way for bacteria cells to communicate with other nearby bacteria cells. And it kind of gives them the ability to determine when there's a certain amount of bacteria cells near them or that they're surrounded by. And then that's going to, this thing called quorum sensing is going to allow them to kind of coordinate what they're doing. So these bacteria cells will all start doing certain things at around the same time, which is pretty cool because these bacteria cells, these are just very simple cells. But quorum sensing allows them to get an idea. It basically tells these cells how many other bacteria cells are around them, if there's a lot of bacteria cells around them or if there's not a lot of bacteria cells around them. And depending on the amount of cells around them, it's going to cause these cells to start doing certain things or not doing certain things. And it kind of creates a coordinated effort where all these cells will start doing something or will not start doing something. So they're kind of communicating in a way that makes what they're all doing kind of in sync together. And so the way this works is you have these bacteria cells, these certain types of bacteria cells that can release these signaling molecules called autoinducers. So autoinducers are the ligand that's being produced in this situation by these bacteria cells. And when there's not a lot of bacteria cells, so these bacteria cells are releasing these autoinducers, and when there's not a lot of bacteria cells, if there's a low density of bacteria cells, there's not a lot of them, there's not going to be a lot of autoinducers around. So there's going to be a low concentration of autoinducers. But when, let's say you have a situation where there's lots of bacteria cells, a high density of bacteria cells, all of those bacteria cells are going to be releasing these autoinducers. And so if there's a lot of bacteria cells and they're all releasing these autoinducer signaling molecules, there's going to be a high concentration of autoinducers in this environment, in this area. And those autoinducers can bind to receptors in these bacteria cells. So these bacteria cells have receptors for these autoinducers. And if there's a lot of bacteria cells, there's going to be a lot of autoinducers. And if there's a lot of autoinducers, then these bacteria cells are all going to have a lot of their receptors becoming activated. Whereas when there's a low concentration of autoinducers, because there's not a lot of cells, these receptors, very few of them are going to be activated. And so what happens is once a certain proportion of these receptors become activated, it's going to trigger a certain cellular response. So let's say in the example that I kind of go through, or the example that I bring up in these notes is the ability of certain types of bacteria cells to become bioluminescent. They start to glow. But these bacteria cells, they all start glowing at the same time. And they only start glowing when there's a high concentration of bacteria cells, which that would be a situation where it's now worth it to start glowing. Like if there was only a few different bacteria cells are microscopic. And if there's a very small amount of bacteria cells, if they start glowing, other living organisms, like multicellular organisms, like animals, for example, they're not going to even be able to, their eyes won't even be able to detect a couple bacteria cells glowing. Like that's not even going to be enough light for our cells, our eye cells to even recognize. And so these bacteria cells, they're actually kind of smart. They will only start glowing once there's a super high density of other bacteria cells. And so when there's a high density of bacteria cells, there's lots of them, thousands and thousands and thousands of bacteria cells nearby, then they all start glowing at the same time. They all start turning on the glowing gene at the same time when there's a lot of them. And then that would be a good situation because now that glowing will actually be something that can be detected by other organisms nearby. The whole point of them glowing is for other things to see them. And now if there's a lot of them, then that's a certain amount of light that actually would be detectable by other animals, for example. And so again, the way this works is there's these bacteria cells releasing these autoinducer molecules. And as there's more and more bacteria cells in the environment, there's more of these signaling molecules that are being produced, these autoinducers. And then these autoinducers start binding to more and more receptors in these bacteria cells until they reach a certain threshold. So let's say like in order to turn on the glowing gene, you have to have like 80% of your receptors becoming activated. And once 80% of your receptors are becoming activated, then it turns on the glowing gene. And what's going to happen, since they're all doing the same situation and they're all in the same environment, is once one of these cells has met the threshold, it's very likely that all the other cells are also reaching the threshold at the same time because they're all in the same environment. So once there's enough bacteria cells and enough autoinducers being released in this area, and it starts triggering 80% of these receptors to be activated in this cell and 80% of these receptors to be activated in this cell, all of a sudden, all these bacteria cells are going to meet the threshold needed at the same time, and then all of a sudden they're going to start doing something together at the same time. Like in this example, now they're going to start glowing. So that's what quorum sensing is. It kind of allows these bacteria cells to get an idea of how populated other bacteria cells, how high the population density is of other bacteria cells near them, so that they can coordinate what they're doing dependent on that. And so this would be an example, again, of paracrine signaling. It's also, you could argue, an example of autocrine signaling because these signaling molecules can also bind to the receptors in the same cell. So some of these autoinducers were released by a certain cell and some of them bind to the same receptor of the cell that released it. So in this situation, we would have autocrine signaling, but also paracrine signaling because a lot of these signaling molecules are binding to receptors on other nearby cells. So both autocrine and paracrine signaling is what's going on here. And it's kind of like morphogens in that you can kind of see some similarities here because both in the case of morphogens being released by embryonic cells and quorum sensing with these autoinducers being released by these bacteria, you have a situation where these cells are responding dependent on the concentration of the signaling molecule. When there's a certain concentration of signaling molecules present and a certain amount of their receptors becoming activated, that's what's going to determine what response happens in these cells. So hopefully that makes a little bit of sense. And then I have a video linked in these slides that kind of show the glowing example, which is a pretty cool example of these bioluminescent bacteria that live on a certain species of squid, actually causing the squid to glow, but it's actually the bacteria that are glowing. And then one last short distance example that is very common is an example involving our immune cells. So in mammals, we have a special type of immunity called adaptive immunity, which involves basically our white blood cells. So in mammals, we produce these specialized white blood cells that can basically adapt to certain pathogens, certain bad guys, invaders that are entering our body. So when there's certain types of bacteria or viruses or fungi or just cells that are in our body that aren't supposed to be there, we call those pathogens. And our white blood cells can adapt to that and formulate a specialized attack targeting that specific type of invader. It's pretty cool. And so what happens in this situation, this is just kind of like a quick little lesson on adaptive immunity and how it involves this type of signaling, this short distance signaling that we've been talking about, is there's these certain antigens. So an antigen is basically a substance that triggers an adaptive immune response. So it's some kind of substance that enters your body that then your immune system basically it activates your immune system cells to then start targeting that substance. That's what an antigen is. It's just a vague term for anything that's in your body that's not supposed to be in your body, basically, and triggers an immune response. And so a lot of times these antigens come from pathogens, these invaders like bacteria, viruses, and fungus that aren't supposed to be in your body. And what happens is you guys have these specialized white blood cells called helper T cells. So the helper T cells are a certain type of white blood cell that you guys have in your body, and they have these receptors for antigens. So they have these special protein receptors that have a specific shape that can bind to specific antigens. And in your body, you're making millions of different types of helper T cells. So you have all these different helper T cells with unique and slightly different antigen receptors. And what's happening is right now in your body, these guys are just circulating your body right now. And then what might happen is when a certain antigen gets in your body, so there's a bad guy, a pathogen that gets in your body, you have these other white blood cells, which are these special type of macrophages, these large white blood cells that go around and basically engulf bacteria cells and viruses, these pathogens. They basically go and engulf them. And then in their cells, they break apart those pathogens into tiny pieces. And then they actually take those tiny pieces and they display them on their cell membrane. So they'll take these tiny pieces, which are now the antigens. These are certain molecules from the pathogen that might trigger an immune response. And they present those on their cell membrane. So we call these special white blood cells, we call them antigen presenting cells. And that's their job, is just to go around engulfing these bad guys and then presenting pieces of them like, look, here's what I found. On their cells. And then what happens is these helper T cells, they can then bind to these antigen presenting cells. So these white blood cells and these white blood cells, they find each other. And if it's a match, if this antigen receptor can bind to this antigen, it's going to initiate a special form of cell communication and signaling that takes place that causes certain things to happen in your immune system. And so what happens here, like let's say we have this situation here. Here's the helper T cell. It has this antigen receptor, and it can bind to this antigen that's being presented on this macrophage, this antigen presenting cell. There's this bad guy, and it breaks it up. It presents a piece of it. This T cell's floating around and binds to it, and it's like, oh my gosh, I can bind to that antigen. My antigen receptor is a good match. What happens then is it triggers the antigen presenting cell to release these signaling molecules called cytokines. These cytokines are these special signaling molecules that get released by your white blood cells and then can activate other white blood cells. So these signaling molecules get released and it actually activates this helper T cell. And then this helper T cell then starts releasing its own cytokines, which can even start binding to receptors in its own cell. So these cytokines can go from this white blood cell to this other white blood cell, and then it can cause this guy to release its own cytokines, which can activate other cells, but even itself as well. And what those cytokines do, those cytokines tell these cells to start proliferating. Proliferating means it's telling the cell to divide rapidly. It's saying, okay, you go ahead and do cell division over and over and over and over again. Make lots of copies of yourself. So then this helper T cell that's now being activated by these cytokines is going to start replicating very quickly. And make copies of itself, because he right now is very important in your immune system, because he has the special receptor that can bind to this antigen. And so he makes lots of copies of himself, and then he goes and activates other special immune system cells that you guys have. And then all of that is going to now initiate an attack on that specific bad guy. So this bad guy that this antigen presenting cell found, now all these guys are going to start going and looking for them in your body to basically target them to be destroyed. And so that's kind of a detailed overview of what's going to happen in your adaptive immune response. But anyway, in this example, when this antigen presenting cell is activating this helper T cell, we actually have three types of cell signaling going on here. We have paracrine signaling taking place, and we have juxtacrine signaling. So through direct contact, there's something happening that causes these cytokines to be released, which can diffuse to this nearby helper T cell. That's paracrine signaling. And then you even have some autocrine signaling happening here with the helper T cell releasing its own cytokines, activating itself. And so you have juxtacrine, paracrine, and autocrine in this situation here. Now, the last type of cell signaling that is in this notes is it's talking about long distance communication. So how cells can not just communicate with cells that are nearby, but they also can communicate to cells that are far away in a multicellular organism. So in your guys' bodies, for example, and in multicellular organisms like plants and animals and other eukaryotes, we're made up of trillions of cells, and there's a lot of situations where cells are far away from other cells. So like the cells in my brain are far away from the cells that make up my different muscle tissue or things like that. And so whenever you have cells from different cell types in your body communicating with each other, that's going to involve long distance communication. Like we're going to now go very far on a cellular level. We're going long distance, not just the cells that are nearby, but to cells in a completely different organ or a different type of body tissue, whether it's like your heart cells communicating with your liver, or your liver cells communicating with your blood, or your brain cells communicating with any other organs in your body. That's all examples of long distance communication. And we call that endocrine signaling. Endocrine signaling is long distance communication that involves signaling molecules being secreted into your blood, that they then can, in your blood, travel to other types of body tissues and affect different types of cells in your body. So basically, anytime you have a situation where signaling molecules are being released into the blood, that's going to be endocrine signaling. That's going to be long distance. Now we're in the blood. Via the blood, we're now going to a different types of cells or different organ or different body tissue in the living organism. Now, anytime you have signaling molecules released in the blood, those are called hormones. So hormones is the term we use for a signaling molecule, a ligand, that specifically travels in the blood. Any ligand that travels in the blood, we call that a hormone. In any situation where you have a hormone, a signaling molecule traveling in the blood, we call that endocrine signaling, which is long distance. And so there's lots of examples of this. You guys have a whole body system actually dedicated to long distance cell communication. It's called the endocrine system, which is made up of these different glands in your body. And these different glands are where certain hormones are being produced. So these glands produce hormones and release them into your blood that then affect different organs and body tissues in your guys' cells. And so we've talked about a couple of these examples already. Insulin's a common example that comes up where in your guys' your pancreas, you release insulin into your blood when your blood sugar levels are high. And so the pancreas makes this insulin, the hormone, the signaling molecule, and it releases into your blood. And then in your blood, that causes your liver cells and your muscle cells to start taking in sugar from your blood. So it tells your liver cells and muscle cells, for examples, and even your fat cells to start transporting glucose from the blood into their cells. So we would call that endocrine signaling, because again, you have an organ releasing signaling molecules into your blood, affecting other organs like your muscle tissue and your fat cells and your liver, which is then going to start removing that sugar from your blood. And then another example I have on here, just another type of, there's lots of different types of hormones. Another type of hormone is called the human growth hormone, or just growth hormone for short. And this is a hormone that's produced by your pituitary gland, which is in your brain, and it gets released into your blood, and it tells lots of different types of cells in your body to start dividing, basically, which is going to allow you to grow and allow certain types of body tissue to grow inside your body. And so the human growth hormone, when it's released into your blood, it will travel in your blood to your muscles, and it tells your muscle cells and your bone cells to start dividing more. It also tells your liver cells and your fat cells to release glucose into the blood so that these cells now have energy as they start dividing, which basically then causes you to start growing, as the name suggests. But again, anytime you have a situation where a signaling molecule is being released into the blood, which would be a hormone, that is long distance communication, or we call that endocrine signaling. Anyway, that's it for these notes and these different examples. So hopefully you guys have a pretty sound understanding of what juxtacrine signaling is, paracrine signaling, autocrine signaling, which are all kind of like short distance communication, and how that's a little different than endocrine signaling, which is more long distance. And that's it for this video. Thank you guys.